Let's discuss about plasmolysis. Before we begin, you need to understand the concept of diffusion. Diffusion of both water and solutes means the movement of molecules will be from a region of higher concentration to a region of lower concentration. If we dissolve a small quantity of solute in a solution, the particles of solute will be evenly distributed eventually. If we consider the example of plasma membrane, the water molecules, being polar in nature, cannot freely cross the plasma membrane. There are specialized channels called aquaporins which facilitate the movement of water molecules from outside to inside the cell. Similarly, the solutes such as ions and sugars are also polar in nature, and therefore they are unable to cross the cell membrane. To help their movement, there are specialized proteins which mediate their transport from outside to inside the cell. Now, the solutes present inside the cell membrane interact with the water molecules, and the water forms hydration shells around the charged solute molecules. As you can see, when the higher solute concentration is bound by the water molecules in hydration shells, there are less free water molecules inside. Thus, the water flows from outside to inside the cell across the concentration gradient. Now, if there are two solutions with unequal solute concentration, the solution with the higher solute concentration is called hypertonic, whereas the solution with low solute concentration is called hypotonic. However, if the solute concentration of the two solutions are equal, we call it isotonic. Now, let's consider an example. If we place the red blood cells in a solution where there is higher solute concentration towards the outside of the cell as compared to the solute concentration of the inside, the water diffuses outside the cell, causing them to shrink. Whereas if red blood cells are placed in a hypotonic solution where the solute concentration of the solution is less than the solute concentration of the cytoplasm, the water diffuses into the cells, causing them to swell up. The red blood cells swell up to the extent that they eventually burst. If we consider the example of plants, they can withstand higher internal pressures because they have a strong cell wall outside. In a hypertonic solution, the cell membrane shrinks within the cell, but the cell wall remains intact. This is called plasmolysis. Now, if we place the plant cell in a hypotonic solution, the cell walls will swell but will not burst because they are strong enough to withstand the turgor pressure. If you want to see the complete video of this lecture, that too absolutely free along with thousands of MCQs, please download our app Hippo e-learning program. And don't forget to like, share and subscribe to this channel.